Alright, this is AP Biology, Chapter 12, the Cell Cycle Lecture. So first of all, cell division is going to be essential to perpetuate life, provides for development, growth, and repair for multicellular organisms, and reproduction for unicellular organisms. Cell division is going to create genetically identical daughter cells. Binary fission is going to do this in prokaryotes, and in eukaryotes it's going to be mitosis, which is nuclear division. So for mitosis in eukaryotes, it's going to be a part of the cell cycle which includes prophase, prometaphase, metaphase, anaphase, and telophase. Alright, so here we have G2 of interphase. So interphase is also part of the cell cycle. You can see some of the events going on during G2. You've got your centrosomes. In your animal cells, the chromosomes have already been duplicated. And after G2, we're going to enter into prophase, which here would be the beginning of mitosis. So the centrosomes are going to start to migrate to the opposite ends of the cells. You'll see spindle fibers start to form. These are going to be made of protein filaments. Then in prometaphase, you start to see the chromosomes become a bit more like what you would normally see replicated chromosomes as. And you can see how the spindles are starting to connect to the kinetochore of the sister chromatids. You can see that the nuclear membrane is starting to break down. Next we have metaphase, and here the chromosomes are going to be lining up along the metaphase plate. You can see how the spindle fibers are again connected to the kinetochores. And once the spindle fibers begin to shorten, they're going to go ahead and start pulling apart the sister chromatids. And they are going to be moving towards the opposite ends of the cell. Prior to metaphase finishing and anaphase beginning, there's going to be a checkpoint which makes sure that all of the kinetochores are attached to spindle fibers. After anaphase and the chromatids move apart, telophase and cytokinesis can begin. Telophase, we see the nuclear membrane reforming, the chromosomes become uncoiled, and we start to get, in this particular case, we've got an animal cell going through a cleavage furrow, and we'll talk about cytokinesis in a moment. So the cell cycle does include interphase, which is made up of G1, S, and G2, and then the M phase, which is made up of mitosis and cytokinesis. The cell cycle is going to be regulated by signaling molecules, cyclins and CDK, which is cyclin-dependent kinases. These are going to combine to form the MPF, which is the maturation promoting factor, or the M phase promoting factor. Alright, so the structure of a replicated chromosome, you're going to have the centromere, the point at which the chromatids are attached to each other. And then the kinetochore is going to be the point on the centromere where the spindle fibers will attach to the chromosomes. And so here we see our two sister chromatids. Here is the centromere region, and here are some spindle fibers that are attached to the kinetochore. And in this diagram over here, we have this centromere with the kinetochore blown up a little bit. And basically what happens is these two red arms here, they're going to start to move back and forth. And as they do, they start to break up this spindle fiber. And it basically kind of chews its way back along that microtubule and separates these sister chromatids. Alright, so the events during the cell cycle, during G1, the cell is going to grow, prepare for replication of the chromosomes, and the cell is going to be metabolically active. There will be a checkpoint here between G1 and S, making sure that we have everything that we're going to need for DNA replication. So during the S phase, DNA is going to be replicated, and then during G2, the cell is going to continue preparing for division. There will be another checkpoint at the end of G2 prior to going into the M phase. All right, so the regulation of the cell cycle by cyclins and the CDK. This is going to control the frequency of cell division, and it's going to vary from cell type to cell type. In skin and intestines, this is going to happen very frequently because, again, these are the cells that are going to be used and constantly rubbed off, basically. Cytoplasmic signals are going to control these checkpoints, which, again, I've mentioned are stop-and-go signals. Okay. So for the checkpoints, you're going to have your first checkpoint here near the end of G1, and this is going to be the most important checkpoint. If no signal is given at this particular point to move forward, the cell is going to enter into a non-dividing state called G0. Some cells will naturally do this, like mature nerve and muscle cells, and these cells are going to be types that do not continually divide. Okay, so the mature nerve and muscle cells are fixed. Liver cells will enter into the G0 stage, but they do retain the ability to reproduce if they have certain environmental signals, certain growth signals given to them. So if everything checks out at this G1 checkpoint, the cell is going to then enter into the S phase, where chromosomes are going to be replicated. When we move from the S phase into G2, the cell is going to accumulate the MPF, that maturation factor, and then the cell can pass from G2 into the M phase. All right, the cell is going to make sure that the replication of DNA has occurred properly, and the MPF will then stimulate the fragmentation of the nuclear membrane, and it will phosphorylate proteins and kinases. And remember, when things become phosphorylated, they become more reactive. So this is going to happen at the end of G2. All right, so then we're going to enter into the M phase, and the first part of that is going to be mitosis. All right, so during mitosis, you're going to go through prophase, prometaphase, and metaphase, and that's when the chromosomes are lined up along the middle. At the end of metaphase, prior to entering into anaphase, we're going to have our M phase checkpoint, and this is where the cell is going to receive signals as to whether or not the spindles are attached to the kinetochores. When all kinetochores have been attached to a spindle fiber, the go signal will allow the cell to proceed into anaphase. All right, so taking a look here again, the most important checkpoint is going to be the checkpoint during the G1 phase. If this one goes, the cell will typically continue through and finish mitosis and cytokinesis. If not, it's going to leave and go through the G0 phase, which again just basically goes to that non-dividing phase. And then during the G2 phase, remember, we're going to have the accumulation of the MPF to make sure that we can go through mitosis and cytokinesis. And then we get to our M phase checkpoint. And at this point, we're just making sure that we have all of the kinetochores attached to spindle fibers. If we do, then the cell cycle can continue. All right, so the cyclins and the CDK, again, the concentration of CDK, that cyclin-dependent kinase, that's going to be constant in the cell. All right, what is going to change is going to be the amount of cyclin, and that's going to change and fluctuate during the cell cycle. And it's going to increase as the cell moves through interphase, through that G1S and G2 phase. And as a result, the amount of MPF will increase as well, because the MPF is the cyclin bound to the CDK. All right, so again, we have our graph here. So the MPF activity is going to be increasing as we move through mitosis, and then as we near the end of mitosis, it's going to sharply drop, and that's because we are going to have a drop in the cyclin concentration. So over here we can see that the CDK is going to be constant all the way throughout, and it's the accumulation of cyclin as we move from G1, S, and G2 that will increase that concentration in the MPF. All right, and as we go through mitosis or go through that M phase, the MPF is going to be used to help mitosis along. And then as we get through the second part of mitosis, that cyclin is going to be broken down. And again, that CDK, that protein, is going to be recycled. All right, so external chemical and physical factors that can influence cell division. We have growth factors. One example is PDGF, platelet-derived growth factor. We have density-dependent inhibitors and anchorage dependence. Okay, so these are, again, chemical and physical factors that can influence cell division. So the PDGF, this is going to help heal wounds. This is, again, platelet derived. All right, so you get a cut. This is going to help to grow and repair those cells. Then we have density-dependent inhibitors, and this is going to be the thing that causes crowded cells to stop dividing. Basically, they'll fill the space they have, and then they are signaled to stop dividing. And then we have anchorage-dependent, and this is going to be where the cells have to be attached to something like the extracellular matrix in order to divide. Now, when we're talking about cancer cells, ways that cell cycle is going to be disrupted in these types of cells is basically that these cells are going to be free of control mechanisms like the density-dependent inhibitors and the anchorage dependence. Okay, these cells may be immortal. HeLa cells from 1951, so maybe you've heard of Henrietta Lacks. Okay, so these were cancerous cells and that scientists use to this day. They are immortal. They have been transformed. Okay, so they don't have those mechanisms to stop dividing. Okay, most normal cells are going to stop dividing after 20 to 50 times. These cancerous cells are usually destroyed by body cells, but ones that are undetected may end up forming tumors. So we have benign tumors, and then we have malignant and metastatic tumors, and these malignant and metastatic tumors can also signal other cells to grow in support of them and help in their nutrition. Okay, so the malignant and the metastatic tumors, these are the ones that are going to be most dangerous to you. All right, so the features of mitosis that lead to genetically identical daughter cells. So we have replication, which again takes place during the S phase. And so that's when our 2n number actually increases to a 4n number for a brief period of time. We have the alignment of the chromosomes along the metaphase plate. So all of those sister chromatids connected at the centromere, they're going to line up one chromosome on top of the other in that line, and then as anaphase begins, those sister chromatids are pulled apart, and then the separation of the chromosomes via the spindle fibers that I just mentioned. Remember, the spindle fibers are attached to the kinetochores, and they basically kind of chew their way back down that spindle fiber, separating the sister chromatids, and then ultimately leading to two cells that are genetically identical. All right, then we have cytokinesis, which is going to be division of the cytoplasm, and it happens a little bit differently in plants and animals. In animals, it causes a cleavage furrow. And this is going to be actin microfilaments and myosin, so these protein strands that are going to help pinch in the cell and split it into two. And then in plant cells, we have a cell plate. These are going to be vesicles filled with cell wall material that ultimately fuse together, and they're going to sandwich cell wall material in between two plasma membranes. And so here we can see the cleavage furrow. All right, basically these are, you can imagine like strings pulling in that cell membrane. All right, kind of see it a little bit better down in here. Ultimately we get two new cells that are genetically identical. Then in this electromicrograph here, we've got plant cells, and you can see these little vesicles filled with cell wall material. Again, the vesicles will be surrounded by plasma membranes. And then as these start to fuse, the cell plate structure forms, and eventually that cell plate structure will fuse with the old cell wall, creating two new cells again, which are genetically identical. All right, and then last we've got binary fission. So we've got two types. You've got asexual reproduction by unicellular eukaryotes, something like a yeast. And then we also have reproduction in prokaryotes. Prokaryotic DNA is circular. The origin of replication appears to be anchored to the cell membrane, and so when that chromosome divides, then we divide directly from there into our two new cells. All right, so this has been Chapter 12, the cell cycle.